All right, welcome back everyone for another deep dive.
Yeah, excited for this one.
This time we're taking a look at a really cool paper all about the SCN2A gene.
Oh, yeah. SCN2A.
You know, it's fascinating how just tiny little changes in our DNA can make such a big difference in how our brains work.
Absolutely. And this paper really gets into the nitty-gritty of that, especially for conditions like epilepsy and autism.
Right. So today we're going to try to really break down how these tiny variations in SCN2A can actually lead to some pretty significant neurological differences, like how they affect this protein called NaV1.2.
NaV1.2. Yeah, that's a key player in all of this.
So first things first, I guess we need to get a handle on what exactly SCN2A is doing and why it's so important in the first place.
Okay, sounds good. Let's start there.
Then we can talk about that old idea, the gain of function versus loss of function, and how maybe that's not the whole story anymore.
Yeah, that's definitely going to be a big part of this. This paper has some interesting things to say about that.
And then we'll get into how this study used this really high-tech method to look at a bunch of different SCN2A variations.
Oh, yeah, they use this really cool automated system, way more efficient than the old ways.
And what they found was actually pretty surprising. It turns out these variations can have all sorts of mixed effects, not just a simple on or off.
It's way more nuanced than that, and that's what makes it so intriguing.
Exactly. And finally, we'll wrap up by talking about why this new understanding is a big deal and what it could mean for future research and treatments.
Because ultimately, that's what we're all hoping for, right? Better ways to help people with these conditions.
Absolutely. Okay, so let's get started with the basics. What exactly is SCN2A?
Okay, so SCN2A, it's a gene, right? And what genes do is they provide the instructions for making proteins. In this case, SCN2A gives the instructions for making this protein called NaV1.2.
NaV1.2. Okay, and what does NaV1.2 do?
NaV1.2, it's a type of voltage-gated sodium channel.
Voltage-gated sodium channel.
Yeah, it sounds complicated, but think of them like little tiny gates on the surface of our brain cells, or neurons.
Like little gates. Okay.
Yeah, and these gates are really important for how neurons communicate with each other. They control the flow of these electrically charged particles, called sodium ions, into and out of the cell.
So they're controlling the electricity in our brains.
Yeah, you could think of it that way.
Wow.
So when a neuron wants to send a signal to another neuron, these gates open up in response to changes in electrical voltage. This lets a bunch of sodium ions rush into the cell, creating an electrical impulse that travels down the neuron.
So it's like a chain reaction of electrical signals?
Yeah, exactly.
Yeah.
And that's how messages get transmitted throughout the brain. It's the basis of everything we do, thinking, feeling, moving, all of it.
Wow, so these little gates are kind of a big deal.
They are. And NaV 1.2 is especially important because it's found pretty much everywhere in the brain, both when the brain is developing and in adulthood.
So it's there throughout our whole lives, working away.
Exactly. And that's why when there are problems with the SCN2A gene, it can have such a big impact.
And what kind of problems can happen if there's something wrong with SCN2A?
Well, if the SCN2A gene is messed up somehow, it can affect how the NaV 1.2 protein is made or how it works. And this can lead to a whole bunch of neurodevelopmental disorders.
Like what?
Well, the big ones are epilepsy, autism spectrum disorder, and intellectual disability.
And those are all pretty serious conditions.
They are, and SCN2A variations are thought to be one of the biggest genetic contributors to these disorders.
Wow. Okay, so we've got this gene, SCN2A, that makes this protein, NaV1.2, which is super important for how our brain cells talk to each other. And if something goes wrong with SCN2A, it can lead to all sorts of neurological problems.
You got it.
Okay, so now let's talk about that gain of function and loss of function stuff.
Yeah, the old paradigm.
Right, so for a long time, scientists thought that SCN2A variations caused problems in one of two ways. Either they made the NaV1.2 channel more active or less active.
Right, like a simple on-off switch. Gain of function, or GOF, meant the channel was basically stuck in the on position. Too much activity. Loss of function, or LOVEF, meant it was off or not working properly.
So more activity, bad. Less activity, also bad.
Yeah, it seemed pretty straightforward. And early on, there was some evidence to support this. Gain of function variations were linked to epilepsy, particularly the kind that starts early in life.
Like babies and toddlers having seizures?
Yeah, exactly. See, the idea was that those overactive channels were making neurons fire too easily, leading to seizures.
Makes sense. And then loss of function would be more associated with autism and intellectual disability.
That was the thinking, yeah.
Yeah.
The idea was that if those channels weren't working properly, it could disrupt brain development and communication between neurons, leading to these conditions.
So based on this, doctors could try to figure out what kind of variation someone had and then maybe use medications to either block the channels if they were too active or try to boost them if they weren't active enough.
Right, it seemed like a pretty logical approach at the time.
But it sounds like this new study is saying that it's not quite that simple.
That's right. This study is challenging that traditional view. They're saying that for many SCN2A variations, it's not just a matter of more activity or less activity. It's way more nuanced than that.
Okay, so how did they figure that out? They mentioned using some kind of fancy new method.
Yeah, they use this really cool technique called automated patch clamp recording. It's a much more efficient and precise way to study the electrical activity of cells compared to the old methods.
So what they did was they took cells and they introduced different SCN2A variations into them, right?
Exactly. They had all these cells, each with a slightly different version of SCN2A.
And then they used this patch clamp technique to basically poke these cells with tiny electrodes and measure the electrical currents flowing through those NaV1.2 channels.
And that's a good way to put it. They're basically eavesdropping on the electrical conversations happening between neurons.
Ooh, I like that. So they could actually see how each variation was affecting how those channels were behaving, like how easily they were opening and closing, how much current was passing through, all those tiny details.
Exactly. It's like having a high-powered microscope for the electrical activity of cells.
And this let them study a lot more variations than they could before, right?
Oh yeah, way more. They looked at 28 different SCN2A variations that have been linked to these neurological disorders, plus a few common ones that are usually harmless, just for comparison.
Wow, that's a lot. And they did all that with this automated system.
Yeah, it's super efficient, like way faster and more accurate than doing it all manually.
That's amazing. Okay, so they looked at all these different variations. What did they find? Did the old gain of function, loss of function idea hold up?
Well, that's where things get interesting. They found that for a lot of those variations, especially the ones linked to epilepsy, it wasn't a simple case of the channel being more active or less active. It was a mix of different effects.
Mixed effects. Like what?
So some of the variations would do things like reduce the overall amount of current flowing through the channel, which you might think is a loss of function, right?
Right, less current, less activity.
But then the same variation might also make the channel open more easily in response to certain signals.
So it's like it's doing two opposite things at the same time.
Yeah, kind of. It's not just about the total amount of current. It's also about how the channel behaves in different situations.
Hmm.
And some of these variations were changing those behaviors in unexpected ways.
So instead of just being on or off, these channels are more like dimmer switches with all these different settings.
That's a great analogy. It's not just about the total brightness. It's about how the light changes in response to different inputs.
And that's what makes it so hard to just categorize them as gain of function or loss of function.
Exactly. It's much more nuanced than that.
The paper mentions something called window current. What's that about?
Okay, so remember how we talked about the channel opening and closing in response to changes in voltage?
Yeah, like the gates opening and closing.
Right. Well, window current is this weird thing that happens where there's a small range of voltages, where some of the channels are kind of stuck in between being open and closed. Like, they're not fully open, but they're not fully closed either.
So they're kind of leaking a little bit of current.
Yeah, you can think of it that way. It's like a leaky faucet. And even though it's just a tiny bit of current, it can make a big difference because it makes the neuron more excitable, more likely to fire off signals.
So if a variation increases the window current, that could make someone more prone to seizures.
Exactly. It's another way that these variations can have these unexpected and complex effects.
Wow, so it's like a whole new level of complexity. And were there any patterns in where these mixed effect variations were located on the NaV1.2 protein?
Yeah, actually. They noticed that a lot of the variations that were messing with the closing speed of the channel, making it close more slowly, were located in these specific parts of the protein called domain 40 and the C-terminus.
Domain 4 and the C-terminus. Okay, and why are those important?
Well, those parts of the protein are really important for the overall structure and function of the channel. So if you mess with those areas, it can have a big impact on how well the channel can close.
It's like if you damaged the hinges on a door, it wouldn't close properly anymore.
Exactly. And they found some specific examples of this, like these variations called R1882L and R1882Q, which were both in that domain IV region. They're doing all sorts of weird things to how the channel was opening and closing.
Oh, and what about that G879R variation you mentioned earlier? The one with the leaky faucet effect.
Yeah, that one's really interesting too. So it had this bigger ramp current, which is basically a measure of how much current is leaking through those partially open channels.
So it's definitely leaky.
Yeah. And that increased leakiness was making the neurons more excitable, even though the overall amount of current flowing through the channel was actually lower.
So it's like a smaller faucet, but it's leaking more proportionally.
Exactly. It's all about the subtle details.
Okay, so we've got all these different variations, some making the channel more active, some less active, some doing both at the same time. It's a mess.
It is a bit of a mess, but it's a fascinating mess. And the more we learn about it, the better we'll be able to understand these disorders and develop better treatments.
I hope so. Now, you also mentioned that they looked at two different forms of NaV1.2. Why was that?
Right. So remember how SCN2A provides the instructions for making NaV1.2?
Yeah.
Well, it turns out that the SCN2A gene can actually produce slightly different versions of the NaV1.2 protein, depending on when and where it's being made.
So like different models of the same car.
Kind of. And the two versions they looked at were the adult form, which is the main one found in the adult brain, and the neonatal form, which is more common in the developing brain.
So like the baby version and the grown-up version.
Exactly. And what they found was that for some of these variations, the effects on the channel were actually different, depending on which version of the protein they were looking at.
So a variation might cause problems in the baby brain, but not in the adult brain.
Yeah, or vice versa. It highlights how the timing of when these genetic changes happen can really matter.
Wow, it's complicated.
It is, but it's also really important because it helps us understand why some people with SCN2A variations develop problems early in life, while others might not show symptoms until later, or maybe not at all.
So it's not just about the variation itself, it's also about when and where it's happening.
Exactly. It's all interconnected.
Okay, so let's bring it all back to the big picture. Why is this new understanding of SCN2A and NaV1.2 so important?
Okay, so the main takeaway here is that we need to move beyond that old gain-of-function, loss-of-function model. It's just too simplistic.
It's like trying to fit a square peg into a round hole.
Yeah, exactly. We need to start thinking about these variations in a more nuanced way, recognizing that they can have all sorts of different and sometimes unexpected effects on the NaV1.2 channel.
And that could help explain why people with SCN2A variations can have such a wide range of symptoms, right?
Absolutely. It can help us understand why some people develop severe epilepsy early on, while others have milder forms or maybe experience more developmental issues like autism or intellectual disability.
And it could also help us develop better treatments, right?
Hopefully, yeah. If we can really understand how each individual variation is affecting the NaV1.2 channel, we might be able to develop more targeted therapies that address those specific problems.
So instead of just trying to broadly block or boost all sodium channels, we could be more precise in how we're targeting them.
Exactly. It's all about precision medicine, finding the right treatment for the right person based on their unique genetic makeup.
That's pretty amazing. And I remember you mentioned something about how the age of onset for seizures might be related to the type of functional effect.
Oh, right. They did find a general trend there. It seemed like the variations that were causing seizures very early in life, like in babies and toddlers, were more likely to have those mixed effects, both gain of function and loss of function characteristics.
So those early onset cases are even more complicated.
It seems that way, yeah. But for cases where the seizures started later, or where the main problems were more developmental, like autism or intellectual disability, those were more often associated with loss of function effects.
Oh, it's like different patterns for different types of presentations.
Right. Although there were some exceptions, of course. Nothing is ever perfectly straightforward in biology.
And you mentioned one exception in particular, that D1050V variation.
Oh yeah, that one's a head scratcher. So on the surface, it looked like a gain of function variation because it was increasing the current flowing through the channel.
More current, more activity, right?
Right. But here's the thing. This variation is located in a part of the protein that's thought to be important for interacting with another protein called anchorin.
Anchorin. Okay, and what does that do?
Well, anchorin basically helps to anchor the NaV1.2 channel in the right place on the neuron's membrane. It's like the foundation that the channel sits on.
So if the channel isn't anchored properly, it can't do its job as well.
Exactly. And some scientists think that when you disrupt this interaction with anchorin, it can actually have effects that are similar to a loss of function, even if the channel itself is letting more current through.
So it's like the channel is working harder, but it's not actually getting the job done.
Yeah, that's a good way to put it. It's like a car with a powerful engine but faulty brakes.
Yeah.
It might be able to go fast, but it's not going to be very safe or efficient.
So even though this D1050V variation was technically increasing current, it might actually be causing problems in a way that's more similar to a loss of function.
That's the current thinking, yeah. It's a good example of how we need to be careful about making assumptions based on just one aspect of the channel's behavior. We need to look at the whole picture.
Wow, it's also interconnected. It's like a giant puzzle where all the pieces are constantly moving.
Yeah, that's a good analogy. And that's what makes this research so exciting. We're starting to put those pieces together and get a clearer picture of how these complex systems work.
So, what are some of the big questions that still need to be answered?
Well, one of the big ones is how do we translate all this knowledge about SCN2A and NaV 1.2 into better treatments for people with these disorders?
Right, that's the ultimate goal.
Exactly. We need to figure out how to target those specific functional changes that are causing problems, whether it's too much activity, not enough activity, or some combination of both.
And how do we do that?
Well, it's going to require a lot more research, obviously. We need to develop better ways to screen for these variations to understand how they're affecting the NaV 1.2 channel in different people, and then to design drugs or other therapies that can specifically address those problems.
It sounds like there's still a lot of work to be done.
There is, but I'm optimistic. We're making progress all the time. And with new tools like this automated patch clamp technology, we're able to study these things in much greater detail than ever before.
That's encouraging. Well, thanks for taking us on this deep dive into the world of SCN2A and NaV 1.2. It's been fascinating, if a little bit overwhelming at times.
Yeah, it's definitely complex stuff, but hopefully we've been able to break it down in a way that makes sense.
I think so. And for you, our listener, we hope you've enjoyed this deep dive as well. It's just a small glimpse into the incredible complexity of the human brain and the ongoing research that's helping us understand it better.
And as always, we encourage you to keep learning and exploring. There's still so much we don't know, but every new discovery brings us closer to finding better ways to help people with these challenging conditions.
Thanks for listening. Until next time.
See you then.